So welcome everybody to our webinar featuring Alessandro Ori about the new workflow for deep protein expression analysis in the formalin-fixed tissues. I would like to give you a very short introduction about diagnosis and why we are so excited to have Alessandro here talking today. And at the end, we'll have a Q&A session. Let me describe this shortly. You have a box where you can type in your questions. Actually, better if you type these questions as they come up during the presentation, because then we can collect them and address them in the end. Now, about diagnosis, who are we? So, at the basis of our company is our mission to help scientists to better understand their biological systems. We are certainly a bit biased in the sense that we believe that it helps to understand the composition and the structure and the activation of the proteome to better understand your biology. Now, diagnosis is founded on 3 pillars, I would say. On top, it's really the R&D. Diagnosis develops, invents, and implements the ever deeper and more precise proteomics technology. And we make this available in 2 ways. So experts that do have access to a mass spec themselves can buy our products and reagents and read our support material so they can implement the latest cutting edge proteomics at their own facility. For everybody else who can benefit from better understanding the proteome of their systems, we offer contract research services where we use the tools, the workflows that we have to directly address the research questions that the people have and help them, even though they have no access to a mass spec. Now, one of the beauties of working in proteomics is certainly that compared to affinity-based methods, we can essentially work in all tissues and all species. So if somebody would come to us and ask us, can we deal with muskrat skin? We can say yes, if we worked with skin before, and we know that muskrat is sequenced, and we don't even need to test this in-house because it's a physical method, right? And but there's one gap in the past where people approached us and asked us, can we deal with formalin-fixed tissues? And we had to say no, because we knew that people had done proteomics before in FFPE tissue, but we were not convinced that the depth and the reproducibility and the quality of the data in general would be sufficient so to really turn this into, say, a useful application that we could do in contract research services. And so when we heard from Alessandro that he has established such a sensitive and robust protocol, we were certainly very excited, and it was great to work with him so that we could implement this in-house as well. And we were certainly even more happy to see that also works in our hands, that now, in fact, yes, we can address also FFPE tissues. And this is certainly highly relevant by itself, as you will see from the presentation of Alessandro for the biological applications. But I always find it also interesting to see how, especially proteomics technology, progresses. Sometimes there are disruptive changes, like the transition to quantitative proteomics or the SWAF DRA technology that really enabled us to overcome hurdles that had been there before. But sometimes it's also the combination of many small improvements that together enable you suddenly do things that have not been possible before. And so now I'm very much looking forward to listen to Alessandro's presentation and not only to hear about the technology, but also about the biological application and with even spatial proteomics that he shows here.
Okay, thank you very much, Oliver, and all the Biognosis team to give me this opportunity to present our work. So the work that I'm going to show you today is, it was our attempt to use mass spectrometry-based proteomics to essentially study the organization of the proteome using clinical-derived tumor specimens. And the motivation to do this came essentially from the fact that there have been a lot of studies around that have analyzed the difference of tumor specimens between patients. And most of the studies have been done using fresh frozen material, which is, let's say, directly amenable to some kind of proteomic investigation. but there have been far less studies that looked particularly at the proteome level at what are the differences within a single tumor specimen. And by this, I mean, what are the differences in protein levels between different regions of the same tumor. This is, of course, relevant, particularly to understand tumor biology, but also in a clinical setting, where very often the clinical judgment of a tumor specimen is made by taking a biopsy from a single region of a tumor specimen. And of course, the challenging and the limitation of doing this in a comprehensive manner and in a quantitative manner, so to quantify thousands of proteins across tumor regions of the same tumor, was the availability of material, because the majority of clinical specimens, as Oliver pointed out, they are in a form, which is the formalin-fixed and paraffin-embedded samples, which has always been challenging for proteomic analysis. So the first step that we had to take was exactly to set up a method that would allow us to get into these immense resources that are these FFPE samples that have not only the advantage of being available because they are the standard method of preserving sample in clinical diagnostics, but they also have an exquisite preservation of morphology that therefore enables to distinguish and to microdissect different tumor regions. As I said, one of the major challenges, particularly for proteomic analysis, but also for other kind of genomic analysis, are the chemical cross-linking of the sample. The samples are fixed, and they also have a poor RNA preservation that makes essentially genomic and transcript level based analysis particularly challenging. Of course, we were not the first one to think about it, but and there have been protocol developed in the past, but our idea was really to kind of make perhaps a small step, but to simplify the methods of sample preparation for FFPE sample to then be able to study the intra-tumor heterogeneity. So therefore, as outline of what I'm going to tell you today, I'm going to first introduce a new method for FFPE proteomics that we developed. And I'm going to show you how we use this to analyze both inter and intra-tumor heterogeneity in hepatocellular carcinoma clinical specimens. And again, toward the end, I'm going to show you a higher resolution analysis of a single HCC specimen, where we somehow pushed our approach to the limit to see what we knew we could discover. And so this graph somehow gives an overview of our approach. So we generally start from tissue slices, and from where we collect tumor region using laser capture microdissection. So this is a standard procedure that is available in most pathology institutes, and we essentially microdissect using a laser small tissue areas. I'm going to get back to you what kind of small means. And then we have combined methods which are so-called antigen retrieval methods, which essentially are needed to reverse this chemical cross-linking that I already mentioned to you. And this essentially involves use high concentration of detergent, such as SDS, extensive boiling of the sample, and we also introduce a step of sonication using a machine called Bioraptor, which we found be extremely efficient in solubilizing this heavily cross-linked material. And the major thing that we had in mind was to limit the sample handling, and therefore the movement of sample from tube to tube, to reduce sample loss, so that we would enable us to analyze the smallest possible samples that would give us enough material for a deep proteomic analysis. This workflow, of course, presents challenges that you can already see some of them for classical proteomic analysis. One of these is the high concentration of detergents, such as SDS, which are not really compatible with mass spectrometry. And therefore, we employed initially a method called SP3, which is based on magnetic beads, that has been shown to be very efficient to essentially remove detergent from protein lysate. And we have now developed an alternative method to the SP3 based on acetone precipitation, which also work extremely well, even with the very limited amount of material. And this was the first step to get our proteins and the digested into peptides. And of course, we wanted our approach to be comprehensive and quantitative. And we have demonstrated in our study that with our approach, both of the two, I would say, major ways of quantifying protein nowadays using mass spectrometry, which are tandem mass tag quantification, as well as data-independent acquisition, are compatible with our approach and yield very reproducible result. Just to give you an idea, a brief idea of what I mean by reproducible, when we were setting up the method, we performed a very simple experiment. So we took 2 consecutive tissue slices from the same tumor specimen and performed twice the whole workflow, including microdissection, tissue solubilization, protein digestion, and protein quantification. And what I'm showing here in these 2 plots are the result of a quantitative analysis, which was performed with data-independent acquisition for 2 different specimens, where on the x-axis we have the quantification performed from the first replicate, and on the y-axis the quantification performed on the consecutive slides using the same approach. And the plots contain each one of these plots contains around 4,000 proteins, and as you can see that we can obtain fairly good reproducibility, quantitative reproducibility across the whole dynamic range. And this is, I don't have a reference here, but I can tell you this is almost comparable to what you would obtain using fresh frozen material of a comparable source. So we were quite satisfied with this. And next we wanted to bring this to see if we could use this technology to answer biological questions. And the question that we wanted to ask was to assess the heterogeneity, the tumor heterogeneity at the proteome level in hepatocellular carcinoma. The reason of choosing hepatocellular carcinoma is that there is, of course, a clinical reason, and that treatments for this kind of tumor are still limited, and therefore, if we could provide some kind of insight, would be of extremely benefit for the clinic. But there is also a technical reason, which is hepatocellular carcinoma is one of the most homogeneous tumor from an histopathological point of view. And therefore, we wanted to see whether, even in the context of a fairly homogeneous tumor entity, we could detect instances of proteome heterogeneity. And therefore, we took 5 HCC specimens deriving from different etiology. And for each specimen, we perform 2 kinds of experiments. In the first experiment, we compared tumor and peritumoral tissues, and therefore used laser microdissection to isolate a specimen from the tumor part and a specimen for the peritumoral tissues from the very same tumor. In the second kind of experiment, we took the same sample, the same 5 samples, and in this case, isolated 2 samples of the tumor part coming from different regions. So we use, of course, the surgical margin to orient ourselves, and we microdissected for each tumor one sample coming from the center and 1 sample coming from the periphery in the proximity of the tumor capsule. So the 2 comparisons were then combined in 2 TMT-10-plex experiments, where each pair of tumor and peritumoral sample, or center and periphery, were directly and quantitatively compared. A question that I'm often asked is how much material do we need for this kind of analysis? And I can tell you that on average, we isolated a surface of 40 millimeter square per sample, coming from a 10 micron slice. I know this number can feel quite high, but if you think we are talking about a tissue volume of around 400 nanoliters. And from tissue volume, we can get sufficient material to perform one TMT or one DAA experiment with quite comfortable amount of material recovered. So the first analysis that we wanted to do was, let's say, a classical tumor versus peritumor analysis. And on the heat map on the right here, you have a hierarchical clustering, which is based on the correlation between the 10 pairs of samples that we quantify. The color code indicating green is high correlation, and in purple indicates lower correlation. This heat map is based on around 5,800 protein groups that were quantified with at least 2 proteotypic peptides across all the 10 samples. So at first glance, you can see that there is a major cluster, green, showing here, which is to a large extent formed by peritumoral tissues that are relatively homogeneous among each other, as indicated by high correlation level. While on the left-hand side here, you have a cluster of mainly tumoral tissues, which show lower correlation between each other. There is, however, an exception, a major exception in this heat map, which is represented by this specimen, which is called tumor 1, where we add the tumor part, which was clustering together with the peritumoral specimen coming from the other samples. This was, of course, a bit surprising, and we went back to the clinical record for these patients, and we actually find out that patient T1, or the patient from which the T1 samples was obtained, had received some kind of chemotherapy before the surgery, while the other patient did not receive this kind of chemotherapy. And of course, this is n equal 1, and therefore it's very hard to draw any conclusion. But we found quite pleasing that we were able to explain this unexpected profile by looking at the clinical record of this patient. What we then proceeded from this was to use the fact that all our sample, tumoral and peritumoral, were paired between each other, and therefore we performed a quantitative comparison for each one of these pair individually. This has the main advantage that comparing two tumor areas, or tumor and peritumoral areas in such case, from the very same specimen, allow us to do not be biased for example, fixation artifacts that might be different between tumor entities. So the result of this quantitative comparison gave us a list of proteins that are differentially expressed between tumor and peritumoral region across these 5 specimens. And what I'm showing you here is a heat map that contains 123 proteins that we found to be differentially expressed in at least 2 out of 5 of the specimen that we compared. So these are the 5 samples, and the color code indicating red whether the protein was upregulated, or in blue whether the protein was downregulated in the tumor part. Again, you can appreciate the tumor 1, as indicated by the global correlation analysis, shows a pattern that in many cases is exactly opposite to the other tumors, again indicating that it's outlier nature. But of course, we wanted to try to find out an independent validation whether these protein differences that we were observing were somehow meaningful for the tumor biology. And to do so, we took an independent data set, a gene expression data set that was obtained by one of our collaboration on an independent cohort of HCC. And we compared our proteomic data to the gene expression data. And this is what is performed in this heat map. And as you can see here at the bottom, with the exception of tumor 1, for all the other tumors, we observed a significant and positive correlation between the fold changes that we observed at the protein level and the fold changes that were observed for the very same gene at the gene expression level in an independent cohort of HCC. This, of course, shows that many of these gene expression changes also manifest at the protein level, but also somehow validate our approach because the gene expression data were not only obtained on an independent cohort, but also were obtained from fresh frozen material. And then we checked if whether this subset of proteins would be able to tell us something about the patients from which the specimens are from, and therefore we performed a classical survival risk prediction analysis, where we essentially stratified patients on the basis of the expression of these 123 proteins. And as you can see here, we can clearly separate long, so short survival based on the expression of these 123 genes that are also showing significant changes at the protein level, indicating that these proteins are not only relevant, not only change in the specimen, but they are also relevant for tumor growth and overall patient survival. So having validated our finding using gene expression data, we also asked the question, but is there something that our proteomic data can tell us about hepatocellular carcinoma beyond gene expression? And therefore, we looked specific for protein changes that would not manifest at the gene expression level. So in the scatter plot here now, I'm plotting fold changes that we measure at the protein level. Again, positive values are values higher in the tumor, against the fold changes that were measured at the gene expression level. And we focused particularly in this area here where we color this dot, which are essentially proteins that show significant changes in protein level above twofold, but that show little or no change at the gene expression level. And this is a limited number of proteins, but we noticed straight away that among proteins which are downregulated at the protein level and not at the gene expression level, there was a subset of proteins that were components of the same protein complexes. And this protein complex is the non-dehydrogenase complex 1 of the respiratory chain. We therefore wanted to look a little bit more in the detail of this, and we looked at the generally and globally at the expression of components of the complex 1 of the respiratory chain, and this is what we do in this heat map here. And as you can see that the majority of the protein are of blue color. Again, tumor 1 shows an opposite trend, indicating a global deregulation of this protein complex in hepatocellular carcinoma. And importantly, the gene expression levels show no change, or if something changed in the opposite direction. We extend a little bit the analysis here by also analyzing the expression of the same subset of protein in a series of mouse models that were generously given to us by the group of Alessandro. So these are mouse models which are genetically defined and therefore driven by a specific mutation. And in this case, we analyzed fresh frozen tissues. But what was pleasing to us and also to our collaborators was to see that this difference in the level of complex 1 was reproduced pretty much in the same way, both in the clinical specimen as well as in these mouse models. And we also wanted to validate this a little independently, and we therefore performed from the very same specimen a quantification of mitochondrial DNA to check whether the reduction of complex 1 would also suggest a general reduction of mitochondrial proteins in HCC, and we could show that in at least 3 of our specimens, a significant reduction of mitochondrial DNA that was going in line together with the observed changes at the protein level. But then, so having somehow validated and found new findings in the comparison of tumor and peritumoral tissues, we wanted to move into the intra-tumor heterogeneity, which was our initial aim. And here we therefore performed this second experiment, where I already described to you, where essentially we have pairs of tumor sample coming from the center or the periphery of the tumor for our five specimens, and they were again quantified in a TMT 10-plex experiment. Here I'm showing you the corresponding heat map as before. Again, over 5,000 proteins quantified across all these 10 samples. And what you can see here is that the clustering of the sample is primarily driven by the tumor of origin. Therefore, tumor 3, tumor 1, etc., they all form this cluster together, indicating that the differences between the tumors, and do not forget these were tumors coming from a different etiology, is much bigger than any difference within the tumor itself, which was perhaps not surprising, but again, pleasing to see in our analysis. And but therefore, obviously, this is not what we were interested in. What we were interested in was the differences before the center and the periphery. And therefore, we took the same approach of quantitatively comparing center and periphery for each one of this pair of samples. And this is what I'm showing here. We were able to identify 43 proteins, therefore a smaller number of proteins, that showed significant changes between the center and the periphery of the specimen in at least 2 of the 5 specimens that we have analyzed. So of course here the number of proteins is much smaller. The differences are of course much smaller, but there are detectable and reproducible differences in protein expression between the center and the periphery of the tumor. And what was important to do is that having this information now, together with the information of the previous experiment, we can now draw these little graphs where we essentially depict, if you want, the tumor specimen, and we can plot at the same time both fold changes between tumor and peritumor, as well as changes between the two tumor sectors that we quantified. So the 2 proteins that I picked here, they are not randomly picked, but they were 2 proteins that were linked to HCC biology by previous studies and showed in this previous study to even have some kind of prognostic value. And what I want to highlight you here, that both of these proteins show, of course, differences in the expression between tumor and peritumor, but also show very significant differences in the expression between the tumor center and the tumor periphery, indicating that if someone would perform a biopsy on the specimen and analyze the center and the periphery, would be probably likely to obtain 2 different results, depending on where this biopsy would be taken. And therefore, this highlights one of the benefits of our approach, that is able to indicate such cases of non-homogeneous expression of potential tumor biomarker within the specimen. And as I mentioned to you in the outline, we also wanted to then push a little bit the limit of our analysis, and therefore we take one more additional HCC specimen, and in this case performed a deeper, more fine analysis of the spatial proteome. So in this case, what we took from the specimen, we performed again laser microdissection and took 2 concentric tumor sectors, starting from the tumor center, going toward the tumor capsule. We also isolated in an independent sample the tumor capsule, indicated here as TC, and took as a reference, of course, the peritumoral tissue again. So in this figure, I want also to highlight another benefit of using FFPE and laser microdissection in the analysis of tumor samples, which is the opportunity that we have, since we have a much closer look to the morphology of the specimen, allow us to remove, in this case, a large blood vessel that was located toward the center of the specimen, and microdissect it out and remove it from our analysis, therefore removing a potential bias in the differential expression analysis. I have to point out that this tumor specimen was chosen because there was no observable morphological differences between the 3 tumor regions, T1, T2, and T3. And also, we performed a mutational analysis of a different tumor specimen using DNA sequencing, exome sequencing, and we could show that it was not only morphologically homogeneous, the specimen, but also homogeneous at the genetic level. This specimen was analyzed in 2 independent experiments. Independent as microdissection and all sample extraction was performed independently and quantified in 1 experiment using data-independent acquisition and in another experiment using tandem mass tag quantification. And the result I'm going to show you here are derived from the data-independent acquisition experiment, and they were very much consistent with what we observed independently with the TMT quantification. So in the heat map here, this now shows again the correlation between the samples analyzed. In this case, the analysis we also performed a technical replicate, which means replicated the injection of the same sample, which of course shows very high correlation of this these samples pairs. And again, the hierarchical clustering very nicely and clearly separate the tumor parts, T1, T2, and T3 here, from the peritumoral tissues, and very much difference was resulting from the tumor capsule, which is something that is also apparent from a normal H&E staining. We validated the protein, some of the protein expression profiling, and here you have 2 examples of decorin, which is a protein which is highly expressed in the tumor capsule. So this validation is performed by classical immunohistochemistry, and here is the antigen EPAR1, which shows opposite expression profile, one being enriched in the tumor capsule and the other one being depleted in the tumor capsule. But this, if you want, was some kind of classical analysis, but our focus was to look into the detail of the difference between the tumor sectors, and this is what we did here. So we obtained the protein profile from T1, T2, and T3, the 3 different tumor sectors, and performed a k-means clustering of the protein expression. And we identify a prominent cluster, a most prominent cluster, which is called cluster T2 here, which shows a group of protein that shows progressive increase going from a tumor center up to the tumor periphery. We looked a little bit more in the detail of this, and we were pleased to see that this was not just a random group of proteins, but that we could retrieve groups of related proteins as being coordinatedly differentially expressed within the tumor specimen. And in particular, we found increased level of ribosomal protein toward the tumor capsule, as well as another group of proteins which are extremely relevant for tumor biology because they have to do with cell migration and cell projection assembly. And these proteins were consistently and reproducibly quantified both by the DIA and TMT approach. And we took one of these, RAC1, and we also validated using, again, classical immunohistochemistry staining that you can see here. And the IHC staining nicely recapitulated the increase of RAC1 expression in this very same specimen toward the tumor periphery, validating our approach. And so I would like to summarize what I've shown you so far. We set up a simplified method that allows deep and quantitative analysis of FFPE sample. This method is highly reproducible and is compatible with both TMT and DIA quantification method. We used this method, and we validated, first of all, this method using independent gene expression data, and show that the information that it can provide can go beyond gene expression and reveal differences in cellular architecture in tumor cells that are not apparent using just transcript level data. And they show you that we could demonstrate the existence of tumor heterogeneity in hepatocellular carcinoma, even in specimens that are morphologically and genetically homogeneous. And we believe that this kind of heterogeneity should be taken into account on the development of protein biomarkers. The study was recently came out in molecular and cellular proteomics, and you can see more details about our work in this publication. I will use the last 2 minutes to just give you an idea of in which direction we are trying to use this technology beyond tumor biology. So since a couple of years, I moved my research group to the Leibniz Institute on Aging, And our group focused mainly on identify molecular mechanism of aging using proteomics as a basic technology. And I mentioned already before that these FFPE samples represent an immense resources because there are millions of samples available. And they can, of course, be used to study disease tissue, but we believe that they can also be used to study healthy tissues, particularly across human beings of different age. So the idea here is to use these FFPE samples and under the guidance of a pathology assessment, identify part of healthy tissues that are part of the specimen, very often included in the surgical margins, or as part of the surgical margins, or for example, in samples that come from transplant, and therefore they derive from fully healthy organs. And thanks to the size of these biobanks, it's possible to identify tissues deriving from human beings of different age and obtain proteome profile across different groups of individuals and correlate these protein expression changes. Just to give an idea that this in principle can work, we have obtained a proteome profile using data-independent acquisition from 12 healthy human liver samples. And in this principal component analysis, each dot here represents a sample deriving from an individual, and each dot is colored according to the age of the donor. And as you can see in this PCA plot, along this PCA1 component, we can quite clearly separate the proteome profile obtained from young donors from the one of old donors. And these differences are meaningful because we can go into individual protein, and here I'm showing you an example of relay, a protein which is linked to inflammation, and correlate its expression with the age of the donor. And as you can see, we can obtain a quite nice correlation between the level, the abundance of this protein, and the age of the donor, which very much likely reflect the increase of inflammation, which is a known feature of aging in humans and other organisms. And with this, I would like to finish and get into the acknowledgements. So this project was started when I was still a postdoc in the group of Martin Beck at the MBL in Heidelberg. And most of the work was carried out by a very talented PhD student, which I co-supervise, Katarzyna Bucia. And the whole project was initiated by a close collaboration with a pathologist, Stefan Singer, which had the opportunity to join our lab for over 2 years and really motivated us to move into this direction. And I also have to thank my team at the FLI, in particular Yvonne Heinz, that she performed the human liver analysis, and Joanna Kirkpatrick for the proteomic analysis. I, of course, thanks the National Center for Tumor Disease in Heidelberg that provided us the sample. And I would like to thank Diagnostics and Alexander Schmidt that supported us with the data-independent analysis setup and analysis. And Lars Zender and Daniel Daug for providing us material from the mouse model of HCC. And finally, I would like to thank all of these agencies for the generous funding that allowed this work to be performed. And I would like to thank you for listening, and I'm very happy to take questions, should you have any.
So thank you very much, Alessandro. Yeah, we got a couple of questions, and I tried to sort them a bit according to technology versus biology, perhaps. Okay, some of them are very specific. Just need a second to sort them a bit. Perhaps here a question. So let's start with the technical ones. So what instrument was used for the TMT analysis?
So the TMT analysis was performed on Orbitrap Fusion Lumos using an SPS MS-free quantification method. And it was, of course, I didn't mention it, we we performed offline high pH fractionation, and then analyzed, I think, around 16 fractions on a Lumos instrument, while the DIA analysis obviously was a single shot.
Okay. And the software that was used for the TMT results is a follow-up question.
So for TMT result, we used a combination of Proteome Discoverer and Mascot, as well as in-house written R script to perform the quantification. And for DIA, we use Spectronaut from Diagnostics.
Okay, and then here's a couple of questions related to the protocol, certainly, because I think, people are aware that this is quite novel and they have questions. So what do you estimate is the amount of loss that you still get during the sample preparation in terms of protein amount?
Yeah, this is probably is very difficult question to answer. We can say, we have an estimate of how better we do in comparison to other extraction, but what is the ultimate yield is a bit difficult to estimate. I would say I cannot answer this question precisely.
And also related to the protocol, so did you test slices from different sources? I guess there's slices that are more or less cross-linked and the tested protocol process.
Absolutely. So I think in now, I would say within this project and also other projects, we have measured, I would say, in the region of more than 200 specimens. And there are some cases that specimens that look morphologically okay yield poor results, but I would say that this is happening in less than 5% of the cases in our experience, which is mainly liver cancer, and this might be due to fixation. Unfortunately, this is something that is not at all under control. There is no record of how long samples were fixed. Our colleague pathologist was very honest with us. He said, if it's a Friday afternoon, the sample can sit in formalin over the weekend. If it's during the week, it's just a matter of hours. And but generally, I would say, assuming that this was the problem with those samples, is affecting a low percentage of overall samples. The success rate is surprisingly high, I would say. Okay.
Let's move a bit to the biology. There's more technical question, but I want to move also to some biological question here. So what do you think you finally revealed in the spatial proteomics differences? Is it hypoxia or enrichment of different cell types?
This is a very good question. We look specifically for hypoxia signatures, and we could not find any. One possible explanation could be particularly for those higher expression of RAC1 toward the tumor periphery, could be rather forces and compression deriving from the proximity to the tumor capsule. There are a couple of studies that showed independently in other model systems that, for example, higher expression of RAC1 could be induced by higher forces. But I think this can be only one of the explanation. And what for sure I could say is that, particularly in the analysis of the 5 different specimens, we saw a lot of differences between the specimens, indicating that there are probably multiple factors that can contribute to these differences in protein expression. And regarding the cell type composition, as far as the pathologist can judge, using, and we performed also additional experiment using, for example, Ki-67 staining to see if we have different level of proliferation between the different tumor parts. We could not see anything obvious, let's say.
Okay, thanks. There's more question coming in still related to the protocol, because I still think that's one of the highlights of this study here. So now you and I have mentioned that it was hard before. to deal with FFPE samples? So what are the real challenges in the sample preparation that people encountered before, and what do you think really is then the magic behind it, solving that?
I think part of the magic is for sure to limit the sample handling. So we found that, we particularly using the SP3 method, the whole solubilization is done on the very same tube, and we found this biorapting very efficient because you just place the sample in a buff sonicator and it is completely solubilized. And of course, in the detergent removal steps, either done with magnetic beads or also acetone precipitation, works very well, and it's very efficient. Are, I would say, the major advantage, for example, to other approaches that have been using in the past. For example, using FASP was not very good for us, and we find that these two alternative strategies, either using magnetic beads or acetone precipitation, are very efficient, even if you have, let's say, 1 microgram, 2 microgram of material.
Mm-hmm. Okay. And here, so about the decross-linking, are the proteins in fact decross-linked, or are they digested while they're still cross-linked?
Sorry, can you repeat again?
It's what do you use for decross-linking? I guess the question is how you revert the cross-links, or are they reverted at all chemically?
This we just use extensive boiling in STS concentration, and that this is something that we did not invent, was more or less developed also as a classical antigen retrieval procedure. We have, of course, I don't think we have 100% reversal, and I can say that because if you look at the conversion rate, like PSM, for example, to protein identification is slightly lower than you would have from a fresh frozen tissue. But this, anyway, doesn't seem to affect dramatically the quantitative accuracy. So there are going to be, of course, some peptides that you will not see, you will lose. But the one that you can quantify seems to be very reproducible.
Okay. Good. And then another biology question again. So you mentioned that to identify changes despite a lack of pathological results, or the visible results from pathology, but have you seen also any correlation between any pathology results with the proteomics data?
I think this is a very interesting question, actually. And I think our N number, the number of samples that we have, is a little bit too limited to answer this question. But for sure, for the future, this could be a very important question to answer. For example, to see if whether some kind of pattern of heterogeneity are associated with a higher aggressiveness. My guess would be yes. It needs to be shown, though. But we can now.
Okay, so I think there's still a couple of questions left. We will select them and answer to people where possible. With that, I would like to thank you very much again for this very exciting presentation, and all the attendees here for joining. You will receive the link in a couple of days after the presentation for your reference, and certainly invite you to stay alert for further upcoming webinars that we will bring from time to time. Thank you.
Thank you very much.